So that's how genomics is being used now, is essentially getting a more accurate estimate of relationships among individuals, which allows us to use these phenotypes much more efficiently, much more effectively.
Hi, welcome to the Poultry Podcast Show. I'm your host, Karen Grogan, and today we are honored to have with us Dr. Jack Dekkers. Jack is the C.F. Curtis Distinguished Professor at the Department of Animal Science at Iowa State University. Welcome, Jack.
Thank you. Very, very nice to be here.
Excellent. Jack, why don't you tell us a little bit about your background and your training and sort of what you do in your daily job there at Iowa State?
Okay, yeah, happy to do that. You may hear from my accent. I'm not originally from the U.S. I'm actually from the Netherlands. I was born in the Netherlands in one of the areas that was reclaimed from the sea, 4 meters below sea level. I grew up on a mixed dairy crop farm. We did have also some chickens, so backyard chickens. I actually, when I was growing up, I wanted to become a veterinarian. And in the Netherlands, you go to vet school right out of high school. So I applied to vet school, didn't get in, just like here, it's limited enrollment. So I didn't get in. So I decided, well, I'll just spend a year in animal science at the Wageningen Agriculture University in the Netherlands. But then when I got there, I really enjoyed it. And so I decided to stay. So I did my bachelor and master's, was sort of combined there in the Netherlands in animal science. As I got through all the animal science courses, I really enjoyed the animal breeding and genetics courses. So I honed in on that, and some of my master's projects were in animal breeding and genetics. During my master's, I actually did an internship at the University of Wisconsin in the dairy science department, and then decided that after my master's, I went back there for my PhD. Another driving force for that was that I met my wife during my internship. So that was another very good reason to go back.
Oh, there's a love story in your background.
That's right. So I went back to Wisconsin to do my master's, and all of that work was in dairy science. During my education, dairy cattle genetics, that's the only thing I was interested in. And so I did my PhD in dairy cattle breeding. Then there was an opportunity after I finished to move to the University of Guelph in Canada, which, at that time and still is one of the major forces in our field, animal breeding and genetics. I was employed there as an adjunct professor, assistant professor, funded by the dairy cattle breeding industry, Holstein Canada and the Canadian Association of Animal Breeding, and then also obtained funding from the federal, the Canadian government, their NSERC, sort of the equivalent of NIFA here. And so I stayed there for 8 years, and then there was an opportunity to move to Iowa State, which also always has been a very strong group in animal breeding and genetics. The history of animal breeding and genetics actually goes back to Iowa State. Jay Lush, sort of recognized as the father of modern animal breeding, worked at Iowa State in the 40s through the 60s, and so it was a very strong group, and it got us back closer to my wife's family. So moved here in '97. As part of that move, I also switched from dairy cattle to working on swine, because that's the thing to do in Iowa.
A lot of pigs.
A lot of pigs.
A lot of pigs and chickens in Iowa. So
yeah, that's right. And by that time, I had recognized that genetics is genetics, and whether you do genetics on dairy cattle or pigs or poultry, you apply the same principles. There's some other interesting questions in those other species also, and you can do more, and because the generation interval isn't as long, so there's a bit more opportunity to experiment yourself also. And then when I got here, so as I said, I was primarily was focused on swine, but then I started working with Highline International, who are headquartered here in Iowa, and interacting with the industry here. And as you said, especially layers and turkeys, they're huge here in Iowa. So then I also started working with, particularly with Highline, started working with them to implement genomics, and also started working with Aviagen on the broiler side. Of course, Aviagen and Highline, they're part of the same
same
company.
Parent company.
Yeah, same parent company. So that's how I got into Iowa, where I am at now, and how I got into also working on poultry.
Excellent. So for people that maybe are not in your line of work, let's start with just a really, really basic thing. Genomics is kind of one of those buzzwords that we all hear about. So explain it to us like you're explaining to one of your college students. How is genomics being applied in terms of poultry breeding?
Yeah, so let me start by talking about how breeding was applied before genomics, right, and including in poultry breeding. And in poultry breeding and swine breeding, they're fairly similar. Is that the breeding structure is like a pyramid with at the top you have the nucleus lines, the purebred lines that are kept in high health farms, a lot of biosecurity, because you don't want infections in those herds, because then you wouldn't be able to sell breeding stock from it anymore and maintain the population. But then, of course, as I'm sure most of the audience knows, at the commercial level, and then both broilers and also layers, it's typically a four-way cross. So there are these multiplication phases, the great-grandparents, grandparental generation, parental generation, to eventually get to the commercial chicks that end up in the layer farms and the broiler farms. But all the selection is primarily driven at the top of the pyramid. Okay? So the four purebred lines that eventually feed into that four-way cross, they are selected within these high health farms, using a lot of data that is collected. Initially was collected primarily in those high health farms, and then used to estimate their breeding values, their value as a parent, for quite a large set of traits. In layers, primarily egg production and egg quality, and using the phenotype on the individual itself, and its relatives. Of course, if you select roosters, you have to rely on the performance of the roosters' relatives. And so prior to genomics, that was all based on pedigree. So you had to not just record the phenotypes, but also record the pedigree, so who is who are the parents of the individuals that have the phenotypes. With genomics, the main change that has happened is that instead of relying on pedigree, we basically use what I call genomic relationships. So if you think of hens that are progeny of the same combination of sire and dam, right? So those hens are full siblings of each other. Based on pedigree, the relationship among them is a half. They share half of the alleles, a quarter from the sire and a quarter from the dam. But if you genotype these hens for a large number of markers across the genome, genetic markers across the genome, then you can actually see, well, what percentage of the alleles do they share, or what percentage of the genes do they share. So then we find out that, well, this pair of full SIBs, they share 50% of their genes, but they share 75 or 80%, right? And so the phenotypes of the other full SIB tells a lot more about the breeding value of a more closely related full SIB. So that's how genomics is being used now, is essentially getting a more accurate estimate of relationships among individuals, which allows us to use these phenotypes much more efficiently, much more effectively. You can also think about it as, so we have all these genetic markers across the genome, and we're talking about, 10 to 50,000 genetic markers across the genome that these animals these birds are genotyped for. You can also think about it that you're basically estimating the effect of each of those markers, right? And then you can determine whether a particular offspring has received the good genes or the bad genes, or how many good genes did it get from its father or its mother, right? That's another way to think about that. So so that's how genomics is being used by, and it's implemented in most of the breeding programs in poultry now also.
So in terms of sort of our classic pyramid structure that we think about for our primary breeding companies, with the adaptation of this technology, I would envision that this would allow changes to occur at a much faster rate. We've always had this rule, it takes 5 years from that elite level to get down to, say, the broiler level of those changes are going to occur. So with genomics, how quickly can our industry adapt?
Yeah, so it actually doesn't affect that timeline from what happens at the top of the pyramid to the bottom all that much. What it affects is primarily how fast we can change how fast we can change the top, the purebreds, right?
So the changes up at the elite pure line.
Right.
Okay.
That's right. So they with genomics you get more accurate selection, and you can also do it faster because you don't have to wait for the hens to have egg production. You can genotype them, as soon as they hatch, right? And get an estimate of how good they would be as a potential parent. And of course, that's even more important for the roosters, right? For the roosters, now you could wait until you have daughters that have egg production. That takes a long time. You can also, what most breeding companies would do before genomics is they would look at the production of the sisters of those roosters. But that doesn't tell you a whole lot because the rooster got a different set of genes than those sisters did. So, but then if you can genotype the rooster, you can make an early decision because you know what genes it received, right? And so yeah, especially on the rooster side in layer breeding, where the accuracy of selection was fairly low because roosters don't lay eggs, as we know. And so, especially there, it has helped a lot to speed up genetic gain. And so that's the main change that genomics has given, that the rate of progress at the nucleus level, at the purebred level, has increased. But of course, that will also result then in a more rapid change at the commercial level.
Your changes there are just more pinpoint. So in terms of as you're talking about evaluating that elite level, how many alleles, as you were talking about earlier, how many are they evaluating in these types of selections?
Yeah, it differs a little bit, but typically it's about 50,000 genetic markers. So they basically 50,000 spots on the genome spread across the genome that they're evaluated for. And what has made this possible is basically a lot of it comes from the Human Genome Project, right? The ability to genotype an individual for 50,000 genetic markers across the genome for less than $50.
Yeah, right.
Even 10 years ago, that would have been 1,000 or more.
Right.
Right. So the technology was really developed at the human genetics level, but then provided huge benefits, also making this feasible, cost effective for livestock. Right? And then, of course, if you compare dairy cattle to poultry breeding, the number of animals that are genotyped in poultry breeding is, well, the value of an individual animal is less than that of a dairy bull, right? So, but even in poultry breeding, the cost has come down enough that breeding companies can afford to genotype in their purebred animals and use that for selection.
And in terms of, as we're moving into different production systems, I know that this is an area that you've examined as well. How our industry can select our breeding lines to, improve performance, and as we shift to, say, like more cage-free layer production. How is genomics impacting that?
Yeah, that's a good question, and that's been a big challenge for breeding companies. Is how to make sure that the genetics that they're selecting results in better performance at the commercial level, right? And especially, like as you mentioned, with the wide variety of production systems that we now see, right?
Yes, so we want to fill every shelf in the egg case, right? So we want.
Yeah, that's right. And and the consumers they demand diversity in that, different production systems behind the eggs that they buy, and that's been a real challenge because in these purebred lines, especially layers in particular, they're usually kept in single bird cages because that then allows you to do individual recording, right? And that's what's needed to make selection decisions. And that worked fairly well when most of the commercial production was multi-bird cage production, right? That if you select a bird or genetics that does well in single bird cages, that does pretty well also in a multi-bird cage. But it's quite different when you go to free range, aviaries, winter gardens, and that's what we call genotype by environment interaction, in that the best genetics in single bird cages may not be the best genetics for these aviary system or these free range system, because there's a lot of other characteristics that start to become important, right? I mean, how they interact with each other, and then if top of that, if you get other restrictions on production systems, beak trimming, then interaction of birds with each other becomes even more important, right? And in single cage systems, you're not able to select for that, right? So that's been a real challenge, and on top of that, also the disease pressure. As I mentioned,
it's very different in those production systems.
Yeah, pure lines, they're very biosecure systems, and of course we can't afford to do that at the commercial level. That level of biosecurity is just not possible. So there's a lot of differences between the environment that birds are the selection takes place in the at the top of the pyramid, and what we have at the commercial level. And one way that breeding companies have addressed this is to record phenotypes at the commercial level, right? They're also crossbreds. That's another difference, right? So collect egg production on individual birds or flocks of birds at the commercial level under these different systems, and then use that data to identify at the nucleus level which birds have the best genetics to perform in that environment. But to do that, you have to track the pedigree from the top of the pyramid all the way down, and that's very difficult. And genomics is a solution there, because what you can do is you can genotype the commercial birds, and you have their phenotype, and then that allows you to estimate what's the effect of this genetic marker or this gene on the performance at the commercial level, right? And then you go back to the top of the pyramid, you genotype the purebreds, the selection candidates, and you can determine which birds have the largest number of favorable genes or marker genotypes, and then select those. So genomics has really been a very important contributor there to allow breeding companies to address this challenge of selecting for the commercial environments.
Right. And in terms of, our customers and society is shifting production in terms of, like we were talking, housing systems, welfare standards. How can genomics help us to, select against things that are maybe natural behavior, or select towards things, like, I don't know, Leghorn chickens tend to be, fairly cannibalistic. Can genomics improve that? Because as we put them into these different housing systems, like you were talking about, welfare standards may say you cannot do beak trimming, and that's a management tool that we use, or beak conditioning, to try to minimize cannibalism. So is there potential of this being applied for selecting, for welfare traits?
Yeah, there's definitely that opportunity. But it all comes down to being able to record the right phenotypes, right, on the right animals in the right environment.
Who keeps up with that, and how that gets back to the breeding?
Yeah, and so that's the big challenge, right? Because what should you record, and then how do you? But then the way to get that back to the purebreds, to the selection decisions, is through genomics, right? So genomics is like I wouldn't say the telephone line, but it's really wireless, right? Wireless connection between the commercial production level and individual animals back to the purebred, so that we can. So if we can record the right phenotypes at the commercial level, and then take tissue samples for genotyping, then we can select for these improved welfare characteristics and behavior under these production systems. And there's still a lot of work that needs to be done there. What are the right phenotypes to record?
Right.
And technology can help there also, because with cameras and sensors and so on, there's a lot of opportunity to implement those at the commercial level, to be able to track individual birds and their behavior and how they interact with other birds. So there's a huge potential there to identify new phenotypes that can be recorded at the commercial level, and then through genomics can be used to make selection decisions at the purebred level. But that's a huge task, but there's a lot of people that are, and I know breeding companies are very working hard on that also to address that.
Definitely. And I think something that, you talked about in terms of disease challenges, how can genomics be used to improve the health of birds in the field?
Yeah, and that's actually something that we work on specifically in pigs. I'm involved in a large project with collaborators from Canada and 7 swine breeding companies. Because they face the same challenge, right? They have this pyramid structure also. And there, what we have done for the last 5 years is set up a natural disease challenge. So in a research facility, research barn in Quebec, they brought in multiple diseases. The major diseases, PRRS, flu, myco, and to create an environment where there are multiple pathogens, multiple diseases, and then every 3 weeks a new batch of pigs is entered into that facility, and it's maintained through continuous flow. So rather than all in, all out, which veterinarians like you tell us we should do. Here because we wanted to maintain the disease pressure, right?
Yes.
And so
you need the older generation to infect the new ones.
Yeah, that's right. And but it's a natural infection. It's not controlled, but we're getting very good data out of that. And so that's one way you could collect phenotypes. That you could do the same thing with birds, and Highline actually have done that with Marek's disease.
Right. I know in leghorns that's a big area of research is Marek's disease resistance.
Yeah. So they every generation birds are, and I think mostly pure line birds are put into a barn where they are infected with the most recent virulent Marek's disease virus, and then record things like mortality or morbidity. So that's what we also did in pigs. So that's possible in chickens also.
So just so I understand the pig model. So the pigs that so they're tested for those disease agents, and the pigs that would either recover or the pigs that would stay healthy, then the samples are taken for genomics for those pigs.
Yeah. So they're not. We are monitoring what pathogens are present for the different batches, right? But we're not identifying for individual pigs what they have, right? Because that would be too costly. But we are then recording growth rate, feed intake, water intake also, and then survival and morbidity, health score. So those are our disease resilience phenotypes, as we call them, right? And then yes, we also take DNA samples for genomics, and so we can identify regions of the genome or genes that are associated with survival under that kind of an environment. Okay. The other thing that we are doing there is before they go into this disease challenge, they come from pretty healthy farms, clean farms, and they go into a quarantine nursery. And in that nursery, we take a lot of measurements, including doing immune assays, look at gene expression, look at protein levels in blood. And the idea there is, can we identify things that we can measure on young, healthy animals that is predictive of whether they would be resilient to a severe challenge like that, right? And that's what the breeding companies want, right? Because then if we find an immune test that can be measured on a blood sample, right. That is very predictive, then they can get that sample on their nucleus animals, right?
Right.
And similar things are being
those.
similar things are being looked at in poultry also. Yeah. So along the same line, I'm also involved in a project in Africa. It's funded by the U.S. Agency of International Development, working with university in Tanzania and in Ghana, and this is led by the University of California, Davis, looking at Newcastle disease. Newcastle disease is not a big deal here in the U.S. because we have good vaccines, right?
It's devastating in the rest of the world, though.
Yeah, because vaccination protocols can be implemented, especially if you look at the village chickens.
It's hard to vaccinate the village chickens. Yes.
Yeah. So the project there was focused on can we breed chickens that are more resistant to Newcastle disease virus? And there we also use a natural disease challenge. So what they do is they go to the markets and find chickens that are clearly infected with suffering from Newcastle disease. They check to make sure it's a velogenic strain, and those birds are then used to seed a flock. And then infect the flock, and then evaluating survival and growth rate and so on. And that's also given us some pretty good data on the genetics of being able to resist Newcastle disease.
That would be great. I think there's a lot of, challenges to vaccination in terms of maintaining cold chain and getting them to those small village poultry in Africa and Asia, and a lot of the, countries that depend on that. That would be really fascinating work.
And then especially, it's important, especially for the women in those villages, because they typically manage the chickens, and it gives them some income from selling eggs or chickens, and so it has a huge. I mean, so when an outbreak of Newcastle disease goes to a village, it's devastating. It has a huge impact. So if we can do something to reduce that impact, that would be very valuable.
So if you could predict, where all of this technology would lead in the next, say, 10 years, where are some places where you can see similar work applied within poultry?
Yeah, well, I think in terms of technology, I think one area where we'll see a lot of advances in phenotyping technology, and I talked a little bit about it already. Cameras to track birds, sensors.
Right, sort of this biosensors and precision farming, and
that can lead into making management decisions more precise, right? But also can generate data from commercial birds at the commercial level that can feed back into selection decisions. So I think that's technology that's developing very quickly and is also becoming cheaper and cheaper. So that's technology that's going to have a huge impact. On the genetic side, we still, the chicken was the first, after the human, was the first animal that was sequenced, right? In 2000, the whole genome sequence after in 2004, I believe, if I'm correct. And that was very, very useful to then find these genetic markers and use them for genotyping. But there's still a lot that we don't know about the genome. Where are the genes? What do they do? Especially for traits like disease resistance or disease resilience, right? And so there's a lot of work that is being done right now to get to know more about the genome, right? And use that to make better selection decisions. Right now, it's still terms of selection, it's still a black box to some degree. We have some idea of which markers may be more useful than other markers, but we still don't know how they work, right? Why does it make why does it increase rate of lay or other traits? And so getting to know more about that is going to be the other technology that's going to be very important and developing fairly quickly.
Excellent. Well, we have a few questions that we normally ask around here on the Poultry Podcast Show. What is some advice that you would give someone who's looking to get into your field of animal breeding and genetics? You're definitely a small field. So if there's, what advice would you give students? You probably advise students in your current role. So what kinds of things do you tell them?
Yeah, well, I tell them my history, right? That initially I was thinking of vet school, and that would have obviously been a good career, but I was kind of forced to take a side road, and then decided that was a better fit for me, right? And so I think one advice that I give students is keep your options open, because you never know.
Advice.
Yeah, you never know. What creates that spark in you, right? And a lot of students, they're sort of when you talk about genetics, they kind of shrug, right? Because they think, well, lots of math and yeah, there is a lot of math and statistics that we do, but yeah, I think statistics is everywhere now, right? And so you can't get away from it. And so I think the other advice I give them is that, again, don't look at genetics and animal breeding as a field that you can't make an important contribution to, because there are major contributions you can make, and through genetics you can have a huge impact. If you just think of that rooster that is selected at the top of the pyramid, right? That selection decision that is made, the impact that has down the line. And I don't remember the numbers, but it's millions of eggs.
Big multiplier, millions. Yes.
Yeah. So just the impact that one selection decision can have. That's in some sense a little frightening, but it illustrates that you can have a huge impact in our field. And there's a huge demand. There's not a lot of breeding companies, but they are scrambling to get qualified people to work in their business. So So what I try to share with them is my excitement about the field, and hopefully that sparked something in there. But everybody ultimately have to make their own decisions, but there's definitely opportunities in genetics. So.
Excellent. Do you have a lot of people read, kind of self-help or business type books or resources that you find useful? Any advice, any suggestions for those kinds of things in terms of how you become successful in your field?
Yeah, I mean, I can't give specific examples, but certainly, there are huge resources there, and it's important, especially for our students, and I think for all of us, they're just coming out of COVID, and we're not out of it, right? That's become even more important to use those resources, for our own, well-being, but also, to be successful in this very much changed environment where, a lot of electronics, a lot of Zoom calls, and and the fast pace of development, right? Keeping, you can't keep up with everything, right? So you have to build strategies to select, what to pursue. And I think, and that's probably the most important, that's been the most important for my career, is find your network and collaborate.
Absolutely.
Right? And that's in any field, but certainly also in research. Collaboration is key to having an impact, right? And in companies also, they're teams. They're not individual players. We're in the middle of the World Cup now, and the Netherlands is playing Argentina this afternoon.
Oh, I see you have your orange on.
That's right. You're
ready.
I bleed orange, and so of course, Argentina has this famous player, Messi.
Right?
Yeah. But in the Dutch media that I follow, they say, well, you can't win the World Cup with one player. It has to be a team. And one of the criticisms of the Argentina team is that they rely too much on Messi, and he's great, right? But you need the team.
It definitely important in any profession is your network and the people that you can pick up the phone and call, dependably and even just to bounce ideas off of, like working through research or research problems, or, hey, I'm trying to get through this, or I have a tough case. And it is definitely critical to build that network.
Yeah.
Jack, I really appreciate your time today. Thanks for everyone for tuning in to the Poultry Podcast Show, and we will catch you on the next episode.